Okay, we'll start in just about one more minute. Let's give people just one more minute to trickle in. Okay, um, yeah, let's go ahead and get started. So hi, everybody. Hope you guys have been having a good week. Um, and welcome to today's lecture. We're going to be continuing our discussion of clustering. And so I'll be sort of following up on some of the stuff that Alex started to talk about last week. And just a little disclaimer, I tested positive for COVID last week, so I haven't been feeling great. My brain and my focus have definitely been a little bit affected, but I'm doing a lot better. And so I'm excited for today's class because actually clustering is something I enjoy a lot. I've done a lot of that in my research, so I've been kind of looking forward to doing it in this class. So I think it should be fun. Okay, so in this lecture, we'll continue our discussion of clustering, and we'll talk about different distance metrics that you can use, hierarchical clustering, dendrogram plots, dbScan, and then for the exercise today, you'll get to look at the MNIST digit data set and practice some different methods on that. Okay, so check out the recommended readings. There's a lot of good resources there. And let's just start off with a little recap of the k-means clustering algorithm. So, and just remember kind of the high level of what we're doing. Clustering is the task of discovering unknown subgroups in data. So you can kind of think of these as natural groups or communities. We call these clusters. And we want to partition the data set so that similar items or individuals are in the same cluster or group, and dissimilar items or individuals are in different clusters, but there's a lot that's very unclear about this that we want to make precise. What does it mean for two items to be similar, and what kind of algorithm exactly do we use to partition the data? Okay, so some examples where you might want to do this is, for example, in social network analysis, you may want to discover communities. Or in ecology, you may want to cluster organisms that share attributes into either different species or genuses, etc. And also in genetics, you may want to cluster tissue samples which share a similar gene expression. And actually for a lot of that type of data, ground truth labels are not available. And so clustering methods are very important because my understanding in the standard pipeline is to sort of first cluster the gene expressions and then annotate them with further information. But, you couldn't directly apply supervised learning because ground truth is not available in those types of applications. Okay, so so here is to kind of recap what you guys already talked about, which is k-means. So for k-means, you start off with a collection of data points. So let's say we have n data points, xi, living in little d dimensions. And the goal is to partition the data into a partition of clusters. We'll call the clusters C, indexed by L. Okay, and the mean of cluster C indexed by L will be mu indexed by L. Let's say we want to find K of them. So we want to find the clusters which minimize the total I guess you can call it a couple things: intra-cluster distance, inner cluster distance, within cluster distance. These are the distances inside the clusters. You want to minimize this objective right here, where you're summing over all the clusters, over all the data points in the clusters, the distance of each data point to the mean of that cluster, squared distance. Okay. And so, so you can kind of think of this as kind of a total measure of the total distance within the clusters, and you want to minimize that sort of within cluster variance. Okay, and then one kind of standard approach for doing this is Lloyd's algorithm. And basically with Lloyd's algorithm, you start off with your set of points, and you use some procedure to pick your initial initial centroids. This could be random, or something along those lines. Usually it's random, but it doesn't have to be. But you pick K initial centers, and then until the algorithm converges, at each iteration of the algorithm, you assign each point xi to the cluster CL with closest center mu-L. So once you fix the centroids, each point gets assigned to the cluster defined by that centroid. You know, what is my closest centroid? I'm then assigned to that cluster. And then you recompute the centroids at the end by averaging the points in the cluster, and you just repeat that process until convergence. Okay, so here's kind of a picture. You start with your data, you start with your initial centroids. So first thing you do is make the cluster assignments, assign clusters, then you recompute your centroids by averaging, the red, the green, the blue. Once you've recomputed those, then you update your cluster assignments until convergence. Okay. And so a couple of properties here. K-means is really fast, okay? So this is a runtime of n times k times d times I, where I is the number of iterations it takes until it converges. That's a little bit unknown, but in practice it tends to run very fast. And then k is usually of constant order, so that's usually not too bad. So you can kind of think of this as order n times d, which is really good. So that's one of the advantages of k-means. One of the disadvantages, however, is that it's not guaranteed to converge to the global optimum. So we want to find the partition, the clustering, which minimizes, which is the global minimum of this k-means objective function. This algorithm is not guaranteed to converge to the global minimum, but only to a local minimum. So for that reason, a lot of times people want to just repeat it. So they may run the algorithm 10 times or 20 times with different initial inputs, different initial centroids, and then choose the partition that has the lowest objective value, lowest value for the loss function overall. Okay, and you can show, so you can prove that the total intra-cluster distance doesn't increase at every iteration of Lloyd's algorithm. So that's why the algorithm will converge to a local minimum, just not guaranteed to converge to the global. And in general, of course, the bigger you choose k, the smaller you can make this value. But you want to find a balance there. You want some sort of reasonable choice for the number of clusters, which we won't really go into that a whole lot in our class, but it is a very interesting question And I think you talked already a little bit about choosing K with the silhouette criteria and some other things along those lines. Okay. Any questions on K-means before we keep going? Okay.
I have a question.
Yeah, go for it.
So it's on the initialization part.
On which part?
On the initialization part of the
Yeah.
centroids.
Mm-hmm.
How does it like, is it like an automated thing that assigns your centroids automatically, or how do you
Yeah.
define?
Yeah, there's different procedures for that.
Mm.
Usually the initial centroids are just data points, right?
Mhm.
So you could do that randomly. So you could start off and just pick K of the data points randomly,
Uh-huh.
and those are your first centers. After that
Okay.
first iteration, they won't be the exact data points though, because then you'll be taking the means of the clusters to
Right.
get them, but they
Yeah.
could start off as the data points
Yeah.
and then they could become the means after the first iteration. But it's usually helpful if the initial centroids are kind of spread out, if they're not too close together. And so one popular way to do the initialization is with the k-means plus plus procedure. That's where you just pick a point at random. That'll be one of your centroids. And then pick the next point randomly, but randomly so that the points that are farther away from your first centroid have a higher chance of being picked. So there's a random procedure which will, sort of favor things to be real spread out.
Gotcha.
And that's helpful so that you don't get unlucky and have all of your centroids kind of start clumped together. That would take longer then to converge and maybe not converge to as good of a solution.
Yeah. This might be like a completely round take, but is it possible to give a context? Like if you know that maybe there's some clusters at a particular data point, is it possible to feed that information to the model?
Yeah, sure. Yeah, if you have if you have some kind of prior information
Mhm.
and maybe oh, these points, I'm sure that these are a certain cell type and I really want to make sure that I kind of, you know,
Grab those.
yeah, that I'm going to converge to a solution that's capturing that, then
Mhm.
you could do that. I mean, I think people usually don't do that, but I could see a situation where you might want to.
Okay, thank you.
Is there on like that note, is there like a method to initialize it for us where it'll pick the random like centroid based on distance? More like, is there like a Python method that'll do that for us, or do we just have to process that?
M++?
Yeah, there is. So that's built into Python. So I'm pretty sure that k-means plus plus is one of the options you can choose when you call k-means, and that will do that. That will do
Okay.
that initialization in such a way that tries to spread out the centroids.
Okay, thank you.
Mhm. Okay. All right. So let's sort of keep going here. So we've discussed also some different methods for evaluating clusters. So this is sort of after you do the clustering post-evaluation. And of course, if your data is two-dimensional, you can look at it, which is very helpful. The problem is that most data will not, be so low dimensional. It can be hard. We'll be talking about dimension reduction next week, and so that will be helpful for that context. And then also there's a lot of different metrics that you can compute, like the silhouette criteria and a whole other things like that. There's a whole long list of them, and you can kind of try to optimize some cluster validity criteria, such as the silhouette. And that's a good way to go about choosing K. Okay, so sort of as we said, often most of the time when you're doing clustering, you don't have ground truth labels, but to kind of evaluate the performance of clustering algorithms, a lot of times we do want to kind of run them on data sets where we have ground truth labels just to evaluate performance. So when you have ground truth labels, here's kind of how you can evaluate. There's different metrics that you can compute to see how well the algorithm is doing. And let's go over a few here. So first of all, you have homogeneity, and the homogeneity metric of a cluster labeling, basically, what does this measure? So a clustering result satisfies homogeneity if all of its clusters contain only data points which are members of a single class. Okay, so it's going to look at the clusters that were computed, and it'll look at how homogeneous they are. So this is, of course, a really useful thing to look at, but what you should remember is this does not account for over-clustering. If you just trivially put each data point in its own cluster, it would have a perfect homogeneity score. Okay, so you have to be careful about that. The completeness score is basically the opposite scenario. So a clustering result satisfies completeness if all the data points that are members of a given class are elements of the same cluster. Okay, this is not going to account for underclustering. For example, if you just trivially put all of your data points into the same cluster, it would get a good completeness score, but that would be a bad clustering. So in practice, you kind of want a good homogeneity and you want a good completeness. And one way to kind of combine these together into a single score is through the V-measure score. And so the V-measure is the harmonic mean between homogeneity and completeness. So you take the product and then divide by the sum. And this is very similar. This is like the F1 score that we talked about back when, I forget which lecture it was, but it's very similar to that idea. Okay, so that's a good one to look at, this V-measure score. And then also the confusion matrix. If you have your confusion matrix, you want to want to see big numbers along the diagonal. But one thing that can be a little bit tricky about making this evaluation is that the labels are really only defined up to permutation. So you will run a clustering algorithm, and you'll get classes 1, 2, 3, but it can't distinguish between 1, 2, 3 and 3, 2, and 1. So in your confusion matrix, you may not see things lining up on the diagonal. They may sort of be permuted, so that can make it a little bit harder to look at. Okay. All right. So distance measures. Euclidean distance is, of course, the most basic one and the most commonly used. And this is the distance that you're used to thinking about. And it's defined in this way, as you're all familiar with. Another distance that's sometimes used is called the Manhattan distance or the city block distance. This is the L1 distance between 2 points. And so you can think of it as computed as summing over the coordinates of the absolute value of the distance of those coordinates. And why is it called the city block distance? Because it's basically the distance you would have to travel if you could only take the blocks in a city. You couldn't take the diagonal. Correlation is also a distance that's used often. well, actually, I shouldn't call it a distance. It's a similarity. So correlation is a similarity measure, which is inversely related to distance. So the more correlated two data points are, the more similar they are, which means the distance between them is small. If points are uncorrelated or dissimilar, then the distance is large. But you can flip these things around in an easy way. Another common one is Jacquard distance, which you can compute between two sets. So if you want to compute distances between sets, one way of doing it is to look at the cardinality of the intersection of the sets divided by the cardinality of the union of the sets. So that measures the overlap of the set in terms of a percentage. And then you can, so again, this is a notion of similarity, but you can turn it into a distance by doing 1 minus that. Okay, and then it can be tricky to know what kind of distance to use. And really so much about getting good results is about using the right metric in the first place. That's probably even more important than which algorithm you use is the underlying way that you're measuring distances or similarities. So that's very important to think about. Euclidean distance works well for roughly normally distributed data, where absolute differences matter. Manhattan distance tends to work better with if you have a lot of outliers in your data. And a correlation measure can work well if you don't really want to measure things in an absolute way. So if your absolute values aren't so important, but your relative values are, or the correlations are, that can be a good way to go. And then Jacquard distance is useful for binary or categorical data. Okay, so we're in the remainder of class, we're basically going to focus on two other than k-means, two other approaches for clustering. Hierarchical clustering, which has a lot of different variants, and then we'll talk about dbScan at the end of class. So hierarchical clustering is a whole family of clustering methods, and it's more than just partitioning the data into k different groups, but it organizes the data into a whole hierarchy of clusters. And there's two main strategies that you can do for this. Agglomerative. So in an agglomerative strategy, every data point starts off as its own cluster, and then you iteratively merge ones that are close together. You can think of making a tree structure. And so you keep merging and merging and merging clusters together until everything is merged into one single cluster. Another way you can do it is with a divisive strategy. That's where you start with all of your data in one cluster and you think about how to cut it. And then you proceed to, cut your clusters until everything is in its own, until every data point is its own cluster. We're going to be looking at agglomerative clustering, where every data point starts as its own cluster, and we think about what is an algorithm for merging. Okay, so this is kind of how we're going to think about it. Start with each item as its own cluster, and then link together the two clusters that are the closest together in some sense. And we'll see there's several different ways to define what are the two clusters that are the closest together. But that's what you're trying to do. And then store this information in what's called a dendrogram plot. And so you can see if you start off with data that looks like this, and then you first merge together the points that are really close together, and then you keep merging closest clusters till everything gets connected together. The dendrogram kind of keeps track of this whole process, And so, so there's not like, so the number of clusters changes depending on where you are, what level of the dendrogram you are. So if you kind of look up here, you can see that you've got like 1, 2, 3, 4 clusters. But if you go down to a more granular level, you have more. If you go up to the very top, you have fewer. Okay, and then, so I said in this agglomerative procedure, you're trying to decide, you wanna merge the two clusters that are closest together, but what does it mean for two clusters to be close together? This is a little bit tricky because a cluster is a whole set of points. How do you measure the distance between two sets of points? There's different ways of doing this. So first of all, you could measure the distance between two sets as the minimum distance between any 2 points in that set. That's called the minimum or single linkage. Okay, so you can look for the 2 points, point in A and point in B, that are the very closest together. Or you could define the distance between those two clusters as, the maximum distance between any point in A and any point in B. And that would be called maximum or complete linkage. But you may think that you might be a little bit more interested not in the maximum or the minimum distance between two sets, but in some kind of average. And if that's your way of thinking, then one thing you can do is look at the average linkage, which is just the average distance between A and B. Or you can take the mean of A, take the mean of B, and look at the distance between the means. That's called the centroid distance. Average linkage and centroid distance are kind of similar in some ways, but the average linkage is the mean of the distances, where the centroid distance is the distance of the means. So the order there is different, and the results you get will be different. Okay, and then finally there's Ward's method, which seeks to minimize the total within cluster distance, similar to how k-means proceeds. And I wanted to show you this picture here because I think this is quite helpful. Let me see if I can move that down a little bit. Sorry, I'm on my laptop today without my monitor, and it's harder to see everything. Okay, but yeah, so take a look at this. I think this is really helpful, just to take a look at some toy data sets and see what the results are for clustering with different types of linkage. In this first column, you see how single linkage clustering will cluster the data. And then in here, we see how average linkage will cluster, how complete linkage will cluster, and how Ward linkage will cluster. And so you can see single linkage is really good at crazy shapes. You can have these crazy rings, the real elongated moons, and single linkage doesn't have any trouble, no matter how crazy the shape or elongated the cluster is. But you can see single linkage will, it has trouble though, when you have pathways in between the clusters connecting them together, and it tends to pick out outliers. So in a data set like this, you can see if you do single linkage, it just picks out a couple outliers as some of your clusters, and then puts all of the rest of the data points into one big cluster, which is not super helpful. So it doesn't do so well on these type of blobby data sets that have some overlap in the clusters. And you can see, on the other hand, some of these other methods that are more about kind of the average distance instead of this minimum distance, they can't handle these weird shapes like the rings and the moons. They have trouble with that. They want to put things into kind of like a circle, to a convex shape. But on the other hand, they do better with these blobby data sets that kind of run together. So in general, it's basically impossible for one method to do well on all kinds of data. So it's good to be aware about which algorithms work well under which circumstances. Okay. Any questions on that before we talk about doing this in Python? Okay, well, let's go over, let's go just a few more minutes before we do our break, because I want to get a little bit into the Python stuff. So of course, I'll leave you to look over the details of a lot of this coding stuff on your own. First, we're just going to import what we need from sklearn and scipy.cluster. Okay, and then let's take a look at some examples. So we can make blobs where, let's say we do 100 points. We want 3 centers or 3 clusters, and we want to call, here's the key Python function that you want to use for hierarchical clustering, which is called agglomerative clustering. So first you will define the model, and you'll define the model by specifying what type of linkage you want to use. Are you using, single linkage, average linkage, complete linkage, or ward? I think those are the 4 options you can do with agglomerative clustering. What kind of distance you want to use, whether that's Euclidean or Manhattan, etc., and also how many clusters you want to cluster your data into. So after you define this model, you can then call the model, and you can fit predict on a data set here, X, as we've defined it. And then we can make a little plot here so we can see the data colored by the labels of this clustering algorithm. And this, it's working very nicely. You get a nice partitioning of the data set, but this is a very easy example. So we expect most things will work well here. Okay. And then, as I said, what we see here is we just see the result if I specify I want 3 clusters. And that means that what has the algorithm done? It's done this whole hierarchical clustering, and then it's just zoomed in on the level that corresponds to 3 clusters. But we can also see that whole dendrogram. We may want more information than just a fixed number of clusters. And so you can do that with a dendrogram plot. And looking at the dendrogram plot can also be helpful for deciding on how many clusters you want to use. Okay, so how do you actually do this? There's a function called linkage that returns an n-1 array that contains all of the cluster merging information, which clusters are joined, and at what distance they're joined. And so let's take a look at that. Then after we run the linkage function, we can run the fcluster function to extract the actual clusters from that linkage array. So, okay, so let's see how we might do this. So first we can call the linkage function, specifying that we want to use the ward linkage. And then you can kind of see what this looks like. This is just keeping track of, this is kind of your index for one cluster, index for another cluster, distance between them, and how many total data points there are. And so don't have to worry about this too much. Basically just think, okay, this is keeping track of that whole tree of information that is the dendrogram, and that we can actually plot here, if we call the dendrogram function after we have called the linkage function, it has all of that information. It can then plot it in a dendrogram. And so you can see here, all of the different points are along the bottom. And then this is just showing the tree of how it's merging everything together at the different stages. Okay. And then we can also call the fcluster function to actually specify, okay, give me, we want to specify basically a cut on this dendrogram, a certain level on the dendrogram corresponding to a fixed number of clusters. And so here we can say, all right, if we want to cluster into 3 clusters based on this dendrogram, what do we get? Those will be the predicted labels here. Okay. Okay, so great. So as I said, that's kind of an easy data set. You expect that most algorithms will do well there. So let's look at a more challenging one, which is the 2 moons data set. This is a simple data set, but it's still actually, it's challenging and interesting. So you see this data set in a lot of papers on clustering. So we can start off, making our data, X moons, and the labels Y that go with it. Here's our data here. And then let's say we do agglomerative clustering with complete linkage and Euclidean distance. And we do 2 clusters. And then we fit that to the moons data and take a look at what we get. And you can see that didn't work so well, that it split both of these moons into 2 pieces. And it's kind of trying, you can kind of see why. It's trying to kind of keep this region together because these are very elongated clusters, but it doesn't look right. You can, it's kind of breaking apart these points, which are, close together. So this is not really what we want to see. So, there's different ways that you could get around this. For example, if you did single linkage, let me see if this will mess me up or not. So if you did single linkage, this wouldn't happen. So we can try that. Oops, maybe I was supposed to call it minimum. Try that. No, okay, sorry. I think I messed up the syntax. Well, I think I didn't run everything from the beginning, so that's my problem. Maybe let me just, I'll try it. Restart and run everything. Okay. Okay, so we were on the 2 moons data set. So now let me try if I do single. There we go. So you can see that single linkage clustering, on the other hand, does really well on this data set, but it has other problems associated with it, which so sometimes it's not the best idea. Sometimes it is. Oh yeah, and there's complete. Okay, but one thing that you can do is add in connectivity constraints. So this is kind of a neat little trick. Previously, we joined the clusters based solely on distance, but we can introduce a connectivity constraint where we look at the k-nearest neighbor graph on the data, and we put in the constraint that only clusters that share nearest neighbors can get merged together in this agglomerative process. And so that's what we do here. So we can specify this connectivity by defining a k-nearest neighbors graph on the data with 10 neighbors. And then when we call agglomerative clustering, we can specify the linkage, and we can also specify this connectivity relationship that we have defined. And you can see if we do that, even with this complete linkage method of defining the cluster distances, it's then able to completely get these two moons, which is a really hard shape to get. So that's kind of a neat little trick and something to be aware of that you can play with. Okay, and then here we can see how this can improve certain results. So here, this is just showing the output of these clustering algorithms. If you ask for 3 clusters with the different linkage types, average, complete, ward, and single, without connectivity constraints, you can see with single linkage, it's just kind of picking off these outliers, which is not great. And then you can see with these other methods, it's just, trying to put it into balls, into kind of convex cluster shapes. But once you add the connectivity constraint, then this kind of stops. In particular, this one looks really nice, where it's able to, well, all of these though look better, that it's splitting the clusters kind of in a more natural way, and it's not crossing this area, this really sparse area where there's no data. It's kind of following the data structure here, which is a more natural thing to do. Okay. Okay, so let's take a look at another example, which is let's take our 5-minute break now. I think that's a good stopping point for our break. So it's 8 minutes after, so let's take 5 minutes and come back at 1:13. So I'll see you back in just a few minutes. All right, why don't we go ahead and get started again? Picking back up here with our next example, which is the iris data set. So you may remember that this is 4 features, 150 data points consisting of 3 different iris species. So there are 3 clusters here in this data set, which we would like to find. So first we can load this iris data set, and then our X is our actual data and our Y is our labels. And then we can call the agglomerative clustering function. Let's again use Ward's linkage with Euclidean distance and 3 clusters. And if we fit this model to the iris data, we can then plot the first 2 dimensions of the data and also the colored by the prediction according to Ward's method. So this is the clustering that you get with the method, and we can take a look at the metrics.vmeasure score. Remember, that's kind of the combination of the homogeneity and the completeness, so it's a good overall score, 0.77, which is, kind of average, not great, but not totally terrible, but it's not great. And then we can take a look also at the ground truth labels here, plotted with the first 2 coordinates. And indeed, this is a challenging problem because there's a lot of overlap in those 2 species. So it's hard to do. And so you can see that we're not totally able to separate those species. But again, we're only seeing 2 dimensions. That's another thing to keep in mind. So, and I remember from a previous lecture, we looked at having a better separation in the last 2 dimensions. So if we change this here to, let's say we look at 2 and 3, then you can see this gets a little easier. And one might imagine a scenario where it's difficult if we do clustering based on all of the features. But if we select a subset of the features, sometimes things may get easier. So we can do this with dimension reduction or feature selection. And that's another important thing that we'll talk about later in the class. Okay. Okay, but, in this case where we are clustering based on more than 2 dimensions, which we can't visualize, one useful tool that you can use is something called a cluster heat map. Cluster heat maps combine the dendrogram with a heat map, so you can judge the values of each dimension in a cluster. It's kind of a nice way you can visualize what the values are doing for all of the different dimensions of the data. And Seaborn provides a nice implementation for this. So let's take a look. If we import Seaborn, and then again load the iris data set, and get the labels, then what we can do is call this function, which is cluster map, right? So we're going to call the cluster map function with the iris data. Let's again use average linkage. Do we use average linkage up here or complete linkage? Actually, I think we're doing a different one here, but let's try it with average linkage, Euclidean distance, and you can see, what do you get? You see the dendrogram, it's been like flipped on its side, but you can see it, and the color here corresponds to those ground truth labels of the species, and then you can see what you want to see is a good separation here in the colors. And you see a pretty good separation, but you see a little bit of mix up between the yellow and the brown. as we observed. But let's see here. You can kind of try to look and see what's going on in each feature, how each feature is kind of lining up with this cluster structure, and that can be helpful, right, as you try to do feature selection, etc. Okay. Okay, so let's talk a little bit more also about evaluating the clusters. We've talked about some different metrics like homogeneity, completeness, and V-measure scores. And so here we'll try a couple of different configurations for these metrics. And you can call homogeneity, completeness V-measure, and that will output all of those metrics, which is kind of nice. Again, the most important one would be the combination here, but for this data, they're all rather similar. And so we can take a look. So a lot of times you may want to know, well, for my data, what's the best method to use? And so here we can try it for this iris data with a number of different linkages, number of different distance functions, and then for each model we can apply it to the data, and then we can compute these metrics and print out what we get. And so here you can see what's going on, and you can see if we're looking at the one with the highest V measure, it looks like the second method was the best, and that was model number 2, which was average linkage with Euclidean distance. Okay. All right, so okay, I think that kind of wraps up what I wanted to say for hierarchical clustering. So in the remainder of class, we're going to talk about another method called dbScan. And then, but yeah, let me take any questions before we move on. So I've got a question in the chat. So the highest V measure is the ideal model to use. I would say, yeah, that's, you know, there's different ways of evaluating these things, but definitely between those three ones that we talked about, the homogeneity, the completeness, and the V measure would be, I think, the most important one to optimize. You can also look at accuracy, but again, the accuracy, you have to be a little bit careful in how you define it, because because you might get like a permutation of your labels. So it's not quite as simple as just taking the diagonal along the confusion matrix. Any other questions? Okay, well, feel free to put them in the chat as they come to you. Okay, so so here's kind of the third method that we're going to go into some details about. So we've talked about k-means, and then we've talked about hierarchical clustering, which builds the clusters in this kind of bottom-up fashion. And the last one we're going to talk about is dbScan, which is actually, it's a really cool algorithm, very well established and popular. And it's a method for doing density-based clustering. So it stands for density-based spatial clustering of applications with noise. I never remember that whole acronym, or just dbScan for short. And so this is an algorithm that is based on the premise that the clusters are basically formed by high-density regions, and that points should be in the same cluster if they're connected by a high-density region of data points. And there's two parameters that govern this algorithm. And so to run it, you have to specify these two parameters. The first one is epsilon, a measure for distance between 2 points, and the second one is min points, a measure for the minimum number of points in a cluster. And we'll see exactly how these parameters come in. So here is the procedure. Start by picking a random point in the data set, and then look at how many points are within epsilon distance of that base point that you picked. And if there are more than min points number of points inside that ball of radius epsilon around the point you picked, then you don't consider that point to be an outlier. You consider it to be a cluster point, and you add the points in that little ball around it to a cluster containing the base point. And then you then proceed to move on to its neighbors. So now you have a cluster including neighbor points, and now you check those points. So for each of those points, you make a ball around them of radius epsilon, and then you see how many points you have inside, and assuming that these should be considered cluster points, not outliers, you add those into your cluster. And so basically you just keep growing these clusters. But if you find a point that doesn't have min points number of neighbors inside a ball of radius epsilon around it, then that point is designated an outlier and it's not put into any cluster. This is kind of a nice illustration of how it works. If you want to play with this, so here's just a mixture of Gaussians, and we can kind of watch how it does the clustering, right? So it started off with a point here. Now it's building the cluster, but at a certain point, it runs out of neighbors, and then it has to hop over to a different point. Then it starts growing the cluster over there. At a certain point, it'll run out and have to hop again. And you can see that most of the points are assigned to clusters, but there's a couple here on the edges which were denoted outliers. Okay, let me, hold on, get back to my page. Okay. Okay, so here's how we do it in Python. So we want to import from sklearn.cluster the dbscan function. And so we can once again start off making some blobs. And then when we define the dbscan model, we have to specify those two parameters, the epsilon and the min samples. The epsilon is definitely the trickier of the one to specify because it completely depends on what the distances are in your data set. So you need to kind of have a notion of how your distances scale because this will really vary depending on your data. There's not like some kind of default that tends to work well. It always has to be adjusted based on the distances in your data. Okay, so then we can fit this to the blobs data and then predict what the labels are going to be. And we can take a look here at our scatter plot of the blobs data together with the dbscan labels. And this is the picture that we get. So with those values of the parameters, we actually got 5 clusters, which are the orange, brown, yellow, red, and blue points. And then the purple points are the outliers. So the algorithm classified these 3 purple points as outliers or noise points. Okay, and then we can see a very good homogeneity score, but this is probably because the data was a little bit over clustered, right? It sort of clustered it into 5 clusters instead of 3. So the completeness score is not very good. And then the V measure is in between.
Quick question. So would there
Yeah.
be situations where you'd maybe change epsilon based on what you're expecting range? Like if one of these outliers you want to include in the cluster, you just would change epsilon to like include it. What kind of ramifications would that have on the analysis we're doing?
Yeah. So yeah, great question. Um, so so definitely how you set the epsilon and how you set the min points will affect, how whether you get points that are designated as outliers and how many. It's a little tricky because these parameters do interact with each other. So so you have to kind of think about like the joint effect as you change both of them. Um, but in general, if you think about keeping, let's say we keep min points fixed at 5, or 10, then if you keep that parameter fixed and you increase your epsilon, then at some point, yeah, you wouldn't get any outliers, because if you make that large enough, you're always going to get 5 points within that ball. So if you think of the min points as being fixed, yes, as you increase the epsilon, you would get fewer outliers, and a small epsilon, you would get more. But because you can change the other parameter, things can get a little tricky. Does that answer your question?
Yeah, that makes sense.
Yeah.
And like, I guess in terms of like analyzing the data, I guess I'm just thinking about in the case where an outlier would be like a outlier and how to like try to make sure that the epsilon isn't a value that is excluding data points that we want included.
Yeah, yeah, but I guess I will say that that this feature of dbScan is actually really nice. It has a built, because it's very common to get outliers in your data. It's very common to get points that kind of mess you up, and we saw that was a huge problem with single linkage. Single linkage is also a density-based clustering algorithm, so both dbScan and single linkage have the advantage of being able to handle real crazy shapes. But the disadvantage with single linkage is that it has no built-in way to deal with outliers. So you get a couple of outliers in the data and it messes up your whole clustering. But dbScan has this kind of built-in mechanism of, okay, these points are outliers. I'm not going to consider those as a cluster, and clustering the rest of the data. So it tends to be a lot more robust with respect to outliers. But the downside is you have to actually tune these parameters, and that needs to be done in a thoughtful way.
Okay, thank you.
Yeah. Okay. So let's see here. Yeah, so I said it works well for crazy shapes, and it also works well for clusters of uneven size. The cluster sizes don't have to be balanced. So this example kind of repeats the blob example, but then filters it so that we have 200 points from the first cluster, 50 from the second, and 10 from the third. So these are very unbalanced clusters, and you can see this picture here. But the method has no problem with the unbalance of the clusters. dbScan still works fine for this. Okay. And yeah, as I mentioned before, it works very well for non-convex shapes. We don't have to add in, the connectivity constraint explicitly like we did for some of the agglomerative clustering or hierarchical clustering methods. If we do dbScan on the moons, we see again that this works very nicely. Okay. And then finally, another example kind of illustrating that the method can handle elongated clusters really well is here. We start off with the blobs, and then we kind of apply a transformation to the blobs, which stretches them out. So we multiply each data point by a matrix, which, elongates along a certain direction. And so you can see here, this is the data that we get if we start off with blobs, and then we transform it to elongate it in this kind of diagonal direction right here. So now if you kind of think about the ratio of the width to the length of each cluster, it's about 2.5 times longer than it is wide. And that again is very challenging for a lot of clustering algorithms, which are built upon the idea of minimizing within cluster distances, within cluster variances. Like k-means will have a very hard time here, as will methods built upon the average linkage and Ward's method. But this is where dbScan does very well. So you can see here that it's not having trouble with these elongated shapes. And the purple points are, again, the outliers, and then it's capturing these 3 components. Okay. We can try it for iris. Now that's going to be a little harder, but let's take a look at this example. So once again, let's do dbScan. Again, I'm not going over how these epsilon values were picked. You have to do that individually for each data set. What's a good heuristic? Is there anything I can offer here? I don't know. You might try computing all of the pairwise distances in your data and looking at like the 10th percentile of all pairwise distances in your data. And maybe that's a good thing to try, and then kind of see how many clusters you're getting and adjust from there. So here for the irises, we run dbScan and plot the predicted labels, and you can see that it's not great in terms of the underlying truth, and the v-score we're getting is only 0.61. And this example, I think, is a good illustration of when dbScan fails, and that's where you have 2 clusters that overlap each other, that don't have great separation. And in this case, the method has a lot of trouble, and you might be better off with a different method. Okay, it also doesn't work well with clusters of unequal density. So here's another example with the blobs, where some of the clusters are really dense and the points are really close together. And we have this other cluster over here, which is very, very high variance, very spread out. And it doesn't do well in this case. It doesn't basically adapt to the local density in each region. It's kind of applying a universal standard about what it means for data points to be close together or far apart. And so, it's kind of not able to kind of, adapt to that local density, and it's splitting this big one into many little pieces. Okay. Okay, so just to summarize, the pros here is that it works great for non-Gaussian shapes and really elongated shapes. It explicitly deals with outliers, which single linkage doesn't. And we don't have to specify the number of clusters. I would say this is a pro and a con. That's kind of nice, but it's also kind of annoying. If you know you want 5 clusters or you know you want 10 clusters, then you have to manually adjust the epsilon until you get the number of clusters that you want. But it can also be a nice thing, so pro and con. And then the cons is this sensitivity to the parameters, having to tune these parameters, and not being able to handle clusters with differences in the densities. Okay, so let me see. We've got like 7 minutes left, and I did have an activity for you guys. I know there's not a whole lot of time to do it. Let me see if there are any questions, and then see if we have time for the breakout rooms, or if you guys just want to look at that on your own. Are there any questions about any of this? Okay. I will say there's also there's a lot of other clustering methods we didn't have time to talk about today. Spectral clustering is also a really neat method that we're not going to have time to cover. But if you're interested in the activity on the sheet, there's also a link to the Python page that talks about other methods that we didn't talk about. Would you guys like to go to the breakout rooms for a few minutes, or is that going to be frustrating because you won't really have time to get very far? I'm happy to put you guys in them if you want, but I know that there's not a lot of time.
Yeah, I'll probably look at it on my own later.
Okay,
So.
why don't we do that? I think 5 minutes is not going to be a whole lot of time, but yeah, but you know, if you want to like find a partner, or you can post it on Slack or something. But yeah, take a look at the activity. It's just the MNIST data set that you have already looked at in your homework, and it's just trying some different clustering procedures on the MNIST data set. So it's a good place to practice. Okay, so let's just do that on our own since we're out of time. And one last question we have: how many activities are there going to be between now and the end of the semester? That's a good question, and I don't know. I'm not sure. I would have to look ahead. So sorry. I would say, you know, if you still need to do a few, I would start to do them, because we are getting towards the end of the semester. Okay. All right. Well, thanks everybody. Have a great day, and we'll see you next week.